So my name is Daniel Rodriguez Leal. I'm an assistant professor at the Plant Science and Landscape Architecture department. I worked in tomatoes, so we do a lot of research trying to understand how we can improve them. And so, for example, there is issues with a fungus we call wilt or fusarium wilt that basically chokes the plant. And so we're trying to understand how we can basically tweak genes to make those tomatoes resistant to these diseases. So just last year there were 190 million tons produced of tomato globally. And when you focus on the processing market, California was probably number 1 or 2nd in production. And so that's why our goal is to really be able to deliver those resistances to that market, because then it would elevate our competitiveness against other countries. We do that through both conventional means, like classical breeding, but we're very excited now that we are able to put our hands into new technologies like genome editing. So this is not GMO. Whereas GMO relies on inserting genes, we're not adding any novel genes. We're basically tinkering with the genes that exist in tomato. The idea is to use these tools to basically surgically change the way these genes are expressed. And by doing so, we can manage to generate disease resistance. So technically, these new technologies, specifically genome editing, they could achieve similar outcomes as conventional breeding. The biggest difference is that whereas breeding entails thousands of years process, we're using these new technologies to accelerate the process of evolution such that we can get the same outcomes but at a faster speed. And over the past few years, we've seen that this technology really is very useful and efficient. But in our project, we're using a different strategy where instead of going after one gene or one section in the genome, we're actually going after multiple. And so we're looking for disease resistance, but if the plant also looks dwarf or it looks like it's not flowering well, so that will tell us that the changes that we're doing are not safe to use because they're changing the way the plant grows, right? But we're also going to look at the DNA level. So, we're very interested in understanding if we can push the use of this technology to create very complex modifications that are still highly specific and that they deliver on the expected phenotype, which is disease resistance.